Hello and welcome to Cannaman TV. In this thoroughly enjoyable interview, I speak with researcher, author and emeritus professor at the University of Guelph, Linda Parker. In this interview, we discuss Linda's background and what led her to cannabinoid research, Linda's work with the late Raphael Mechoulam, Linda's work including Cannabis in the Brain and CBD What Does the Science Say, Linda's up-and-coming release, Cannabinoids for Essential Knowledge, the endocannabinoid system and nausea, and the future direction of cannabinoid research. Make sure to like and subscribe, and join me on Patreon and PayPal to help to build a stronger media platform. Now then, let's talk cannabis. Let's say, Linda, thank you so much for coming on the channel. It's an absolute pleasure.
Well, thank you. Thank you for having me, Connor. It's really nice to be here with you.
Excellent,
On this
excellent.
day.
It certainly is. It's an absolute cracker today. So just to get started then, can you give a little background on how you became active in cannabis research?
Oh, okay. Okay. It's actually I had been working on nausea work for quite a while. And in the year 2000, I had an airmail envelope came to my office mailbox. This is one of those old-fashioned, you've probably never seen them before the days of email, old-fashioned airmail envelopes, and it had Hebrew writing on it, which was very strange, unusual for me. So I opened it up, and it was from Raphael Mechoulam, and so he had read this obscure article that Cheryl Limebeer, a PhD student, and I had published in this in a journal on the anti-nausea effects of THC using a rodent model that we had kind of developed for this purpose. So we showed that THC reduces nausea in this model, and there weren't any rodent models available at the time. And nausea is one of the effects that THC is known to produce. It had been approved by the FDA for this purpose. And nausea is a big problem for chemotherapy patients because there's really good drugs to treat vomiting. There's the 5-HT3 antagonists, ondansetron, are really good for vomiting, but they don't really affect nausea. People still feel really sick, but they don't necessarily vomit.
Right.
So there's a need for drugs to treat nausea. And so Rafi, and in this letter that he wrote to me, he had read this article, and he very modestly introduced himself to me as, he said, "I too am a cannabinoid researcher," just like you, which was that was his style. That's who he was. I mean, he passed away not last March. Last March he passed away. Yeah. But he was such a modest person, just a lovely person. And he introduced himself this way, and in the letter he suggested that he send me some CBD, and we test CBD using this model to see if it also reduces nausea, because there was nothing about it, had never been tested for that purpose.
Yeah.
So that started this 20, almost quarter of a century collaboration that I had with him, and he brought me into this cannabinoid field. So CBD did work. Then we tested other compounds of his, they worked. And we published 3 or 4 papers before we even met each other. And then he invited me to come to the International Cannabinoid Research Society. I don't know if you're aware of that meeting, but it's a meeting that he sort of organized, he sort of originated. And all the major players in the cannabinoid field around the world meet once a year at ICRS. Right. And he invited me to come to Monterey, where it was meeting that year. And and I met other cannabinoid researchers, like Roger Pertwee is one of my I saw that you have an interview you interviewed Roger. And,
Yeah, Roger was brilliant. Yeah, it was a pleasure and absolute honor to have him on the channel, 100%.
yeah. So Roger and I started collaborating, and then we connected with all kinds of other researchers, chemists who wanted us to test their compounds for anti-nausea effects or nausea-inducing effects if it's a CB1 antagonist produce nausea. So my students and I kind of became engaged in cannabinoid research. We got pulled away from kind of traditional the neurobiology of learning, which we were doing beforehand, and into this cannabinoid field. And it's been wonderful because the students have had wonderful collaborations with people, and Rafi brought us all into this. And then we continued to collaborate with Rafi on CBDA after, and Roger Pertwee. Erin Rock was my postdoc. She did this work. And she found that CBDA was about 1,000 times more potent than CBD in reducing nausea. Now, neither of these have ever been tested in humans, in human clinical, a good human clinical trial. So Sativex is a combination of THC and CBD, and there are papers published on Sativex as reducing nausea in humans. But CBD alone has never been tested in a good quality clinical trial for anti-nausea effects in humans. But we've known it for 20 years that CBD is effective in reducing nausea, and CBDA is about 1,000 times more potent than CBD. And then, but CBDA is unstable, relatively unstable. It converts to CBD with heat. It's in the raw plant. CBDA is in the raw plant, and when it's heated, it's converted to CBD. So because it's fairly unstable, Rafi Mechoulam synthesized a methyl ester version of CBDA methyl ester, and we found that was even 10 times more potent than CBDA. It's really effective in reducing nausea. So this is some work that I hope gets into clinical trials, because I know that Rafi would want to see this in patients' use with patients. His whole kind of motive for doing much of this work was to try to reduce patient suffering, and that's he was always a big advocate for clinical trials to get some of this basic science out into the clinic to help people.
Yeah, it's fascinating, quite honestly, because with this this archive of work, and for it never to be pushed through a clinical trial. So I mean, one of the things about CBD that popped to mind was Epidiolex. If that's the correct way of saying it. So has that only been tested in a clinical setting for seizures? Is that the reason? Was that
for Epidiolex, it's been developed for seizures. Sativex, which is a combination of THC and CBD, has been tested for a lot of things. For pain, for multiple sclerosis symptoms, and some of the clinical trials with Sativex have suggested that it is effective for pain. And there recently, and just in the 2020s, it's been tested for nausea, Sativex, and it's effective. But we don't know if that's we know THC is effective. We don't know if that's the combination of THC and CBD, or if it's if CBD is playing a role there. And because it hasn't been, there's only one case report out of Brazil that suggests that in this the patient was helped by CBD, but there's nothing in the literature for other than that one case report for CBD.
Yeah. So what is the hurdles then? What's inhibiting the ability for CBD to be tested individually in humans in clinical trials? Do we need private investors, or I mean, is there just, you would think that,
I guess it's always a problem. Clinical trials are so expensive, and I mean, clinical trials are with cannabis are difficult because it's not patentable, and so it has to be analog of some cannabinoid that might be patentable. But I think it's time for NIH to get involved and start funding some clinical trials with cannabinoids to find out if some of these basic effects are translatable into humans in the clinic.
Mm-hmm. It's fascinating. So I mean, why do you think that there's not more focus and attention on studying this? Because I mean, inevitably, as you said, there's like hundreds of molecules to be studied in the cannabis plant. So that's like,
For sure.
is there another plant or that's so worthy of attention? I don't know what's.
I think actually I was at ICRS last year, this International Cannabinoid Research Society. NIH was there, and representatives from NIH were there, and they are focusing now on minor cannabinoids. They're interested in what some of the minor cannabinoids are doing.
Right.
So there's CBD is somewhat of a minor cannabinoid, but it's actually it's one that's been studied quite a bit. But some of the others like CBG and CBDA, THCA, THCV are cannabinoids that have effects that NIH is starting to look at. I mean, some of the work that Erin Rock did as a postdoc in our lab was looking at combinations of compounds. So, for instance, we found that CBD and THC synergize.
Mm-hmm.
So if you give subthreshold doses of THC that don't, and this is in the nausea model, if you give subthreshold doses of THC and subthreshold doses of CBD, doses that don't reduce nausea on their own, when you combine them, you get a synergistic effect.
Right.
So they seem to be enhancing the effect of one another, but by different mechanisms, which is interesting because THC acts as an agonist at the CB1 receptor, but CBD is reducing nausea by acting as a 5-HT1A agonist, a serotonin receptor 1A agonist. So, and these, even though they're different mechanisms, they synergize and they produce a more potent effect. CBDA and THC also synergize. CBG it counteracts the effect of CBD on nausea. Right. So if you give CBD, you get a reduced nausea. If you give CBG, it prevents CBD from reducing nausea. Oh, so these compounds are all fighting against. That's how Roger Pertwee and Rafi would say they're fighting against each other to produce. And so we started looking at some of those effects with GW Pharmaceuticals in Great Britain. They sent us. That's how we got a hold of these different compounds, and they sent us about 12 different compounds from the plant, and we tested combinations of these on nausea, which is sort of how you're going to have to approach the entourage effect, at least at a scientific level, is each of these compounds is having a different effect at a different receptor, in some cases, or the same receptor in some cases, or an opposing effect at the same receptor. And it's this combination of these compounds that produce what people talk about as the entourage effect, I think. Yeah. But there's over 100 compounds. So to ever hope to capture that effect and try to understand it is going to be very difficult, or maybe impossible. I don't know.
It does seem like that there's almost at least a century's worth of research required before there's the fundamental foundation of actually what the interactions are. Because again, also I mean, unless I'm mistaken, there's 126 cannabinoids identified at this moment. Is that right?
Oh, might be. I'm not sure. I can't keep up with that.
That's what I was thinking. So I mean, the chances in my mind, the reason I was asking was I was like, the potential for them being like 200 cannabinoids, is that possible, or are we kind of at almost like a peak where we're aware that there's only this many?
I guess it depends on what if you can include terpenoids and terpenes as cannabinoids, then I mean, it's I'm not sure.
Absolute minefield, yeah.
Yeah,
Yeah.
and I'm
No,
not a chemist. It sounds that's
no, quite right. And the thing is, that's what actually takes us perfectly on the next question. Then your first book, Cannabis in the Brain. What was your motivation for that book, and what was the findings as a consequence of creating it?
okay. Well, actually, that book came out of a review article that Rafi and I wrote. So again, just to explain, just to talk about Rafi's generosity and kindness. He was invited to write a review article for a journal, for a publication called the Annual Review of Psychology, which is a really big deal. It's a big deal for, I'm a psychologist, he's not. I'm his token psychologist. I was a token psychologist. And and so he got invited to write this chapter on cannabinoids in the brain. And so he asked me if I wanted to co-author it with him, which is really kind because it's definitely the highest cited paper I've ever written. There's only one edition of this each year, the Annual
Right.
Review of Psychology, and there's only one neuroscience article each year worldwide that comes out in it. And they asked Rafi to write that. And that was a really big deal. But he was just so kind. And he even wanted me to take senior authorship. And I said, no way. Just way too kind. So we wrote this article on cannabinoids in the brain, and it touched upon some of the things that I touched on in the books. So I had put a lot of time and energy into this, and I asked Rafi if he wanted to write a book. I talked to MIT Press about a book on cannabinoids in the brain, and they were keen on it, interested. They were all gung ho. And so I asked Rafi, and he said, no, I'd rather just work in the lab, and I know book right now. So I went ahead and wrote the book. And so it came from that chapter. That chapter was I had done all of this work and decided why not turn it into a book. I think we need to get the information out beyond these journal articles. So that was really the motivation for it. And I guess then we wrote after that book. I did ask Rafi again if he wanted to work on another book published by MIT Press on CBD, and because there was so much misinformation. There's so much misinformation about CBD and what it does and magical, things. And there's really good science, and there's some really good science suggesting CBD may do a lot of things, but in terms of taking it to human trials, there's very little evidence of anything except for epilepsy. So I asked Rafi, and he said, well, and this was in the early 2020s that we started it. It came out in 2023. He said, sure, let's do it. Let's write this book together on CBD, and with Erin Rock as a postdoc. It was my postdoc at the time. She's a co-author. He said my research, my career began with CBD. He said that my very first paper was on this identifying the structure of CBD in cannabis. And he said, it's fitting that my career end with CBD, which unfortunately it came close to the endings. But I was able, Erin and I were able to work with Rafi on this project in the last, during this period, kind of and it was just amazing. We had emails back and forth every week. And so it was a wonderful experience for us to have with him during that time, pulling that book together. So that was the motivation for that one, just to try to dispel some of the myths and to try to get out what the really hopeful science that's there about CBD.
It's interesting. I actually contacted Raphael on about it was unfortunate, obviously, about 9 days before he passed, and Yeah, and he messaged me back, and so I'd contacted him, asked him, would you like to? 'Cause I messaged him before. It was about 3 or 4 months before. In fact, maybe more than that. I think it was in November, the year before. And then he said, "Yeah, I've got a lot of work on at the moment. I'm not doing many podcasts, but contact me next year around about May time, that time period." So then that time period arrived, and then I'd emailed, and he says, "Let's go on a call." And it was about 9 days before, and he sent me over his number, and I was like, "Okay, I'll give you a call in the next couple of days." And obviously that call never manifested. And it was like 9 days later, obviously, that it was all everywhere, obviously, considering his status and his influence in the world of cannabis. But that was extreme. We lost a legend. I mean, he rightfully so called the godfather of cannabinoid medicine, research, you know. But it's brilliant to hear such positivity about him, because obviously you don't really know much about him on the mainstream. You just see the amazing work that he's produced. And touching back on the first book, the cannabis in the brain, what was the main piece of information that you remember from that body of work that was most profound, or that you found most interesting about creating that book?
Well, I think the most important message that we tried to get out with the review paper and with the book is that I think a lot of people don't know, the general public doesn't know that cannabis, that THC is really the psychoactive substance in cannabis,
Mhm.
and that it acts on a natural receptor that is found in our brain, that actually is there because our brain produces a compound like THC.
Mhm.
Not doesn't act exactly like THC, but that it acts on the same receptors that THC acts on. So that the anandamide story and and 2-AG, the endogenous cannabinoid story, is sort of what I was trying to get out with that book, that not only do cannabinoids do this, but also your brain does this under natural conditions. Stress, for instance, it can reduce uh, anandamide, and if you elevate anandamide, it can overcome the anxiety and depression produced by stress. So it's a natural system that it's acting on, and this is that natural system. So the cannabinoid system of the 90s, discovered in the 1990s, is much like the endorphin system that was discovered in the 1970s, the way that morphine works. Morphine, the opiate receptor, isn't there for us to take the opium poppy
Mhm.
and get high from it. It's there because endorphins are produced. And so if we study how cannabinoids work, what effects cannabinoids have, it gives us a clue into what the natural system is there for, what the purpose of this is. So I think that's the main thing that I was hoping to get out, and I think the main purpose of that review article, was to explain how the natural cannabinoid system works.
So what does that mean then, from the outside looking in? We have an external situation with CBD and THC, and we have an internal system which mirrors that with anandamide and 2-arachidonoglycerol. It's like, what does that mean then? Because obviously, like, does that mean that it's just there's like symbiosis of nature and biology over long periods of time, and this is what we're looking at, or or is this presented to us in order to utilize it in some form? I mean, what does that mean?
What does it mean in terms of finding these plants? I
Yeah.
guess that humans have been experimenting with plants growing around them for centuries. In fact, I think that history of cannabis goes back to ancient China,
Yeah.
at least the known history of it. Maybe it was even before that. But so people have experimented with plants, and those that produce certain effects are ones that they're more likely to use again. I don't know. I'm not a botanist, so I don't know
Mm.
why the
Yeah.
plants are there that affect our brain in a certain way. But I know that humans have been experimenting with different plants to for as medicine
Mhm.
for pretty much throughout their history.
It's interesting. I
So
watched this clip of Richard Dawkins talking about his memes, his meme theory, and this whole thing is that the organism's a representation of the external environment. And I think that's the thing that pops to mind with that. It's almost like we're in a meme competition with nature.
I don't know, maybe.
Yeah, it's quite funny. But yeah, so I mean, so the next book, as you briefly mentioned, there's CBD. What does the science say? That seems to be obviously inevitably cannabis in the brain is an extremely important book, but for the past couple of years in the UK, we've been in a back and forth of what CBD, the role it has, the regulation format that it should be in, methods of dosing and the quantity required, and all these different other elements of discussion around CBD. So what did you find with CBD? I mean, I think there was a few of the things when I skipped through some of the chapters that you were talking about. The medicinal value, so like a neuroprotection, cancer, IBS, things like that. So what could you say? What was the main findings from CBD? What does the science say?
Well, it's kind of that's kind of hard because that book was written by 3 different people.
Yeah,
So
yeah.
I wrote some of the chapters, Aaron wrote some of the chapters, and Rafi wrote some of the chapters. But I think one of the main issues with CBD that is that it, it's often taken orally, and it has really poor bioavailability. So when it's taken orally, it's metabolized in the liver, first pass metabolism, and then there's the same enzymes metabolize it that metabolize other drugs often. So you get drug interactions with CBD, which could be problematical. But it also doesn't get to the brain easily
Right.
because of its poor bioavailability. So I think one of the things that one of the new directions with CBD, one of the things that people are working on is to develop better delivery mechanisms, different kinds of vehicles for CBD to be dissolved in to allow greater bioavailability. So when people are taking it orally, and often the doses that are used by humans are much lower used kind of across the board. That we might get from CBD from the internet, purchasing it from the internet, are lower doses than often the doses that are used in the laboratory, but and injected, for instance. So that I think quite often there's not a lot of CBD getting to the sites that are necessary. And also there's some of the work by some researchers show that where they've assessed the composition of the CBD, that often there's THC in the CBD that's sold over the counter. So quite often people aren't getting what they think they're getting when they take CBD. That's, I think, one of the things that people need to know about.
That's one of the things I found quite fascinating about CBD is because it seems to be, particularly in the UK, that's the introduction to cannabis for the culture. That's for people that are not ordinarily interacting with cannabis as a plant. CBD is the molecule that they've been introduced to. And I think one of the things that's been frustrating about that is that often the quality of product which is being sold in the different varieties of shops, the quantity of CBD is not legitimate. It's not the number that they're saying on the tin. Essentially, CBD has been kind of misrepresented in a lot of ways in the UK, and I think that's what's so important about the book is that it highlights a huge number of elements that CBD can interact with. So the next book you're working on is Cannabinoids for Essential Knowledge. Can you talk a little bit about that?
Okay, well, it's just a small book. It's a series that MIT has for kind of almost informed lay people, for people who want to know about cannabinoids but don't want a lot of scientific detail and all the references and all that. So it's a small essential knowledge about what cannabinoids are, what they do, and so it's sort of a synopsis of the 2017 book, Cannabinoids in the Brain, with an update of what we know now relative to what we knew then. And surprisingly, there's not a lot new in terms of clinical trials. I've kind of been shocked to find out that there's since 2017, not a lot of new clinical trials have been done with cannabinoids. The COVID is probably part of the problem that happened during that period, but so I'm trying to update the information and then just provide it in a more accessible manner about the endocannabinoid system and cannabinoids. It's a short little book, so
Excellent.
I haven't finished it. I don't know. Yeah, I'm hoping I'll get it finished. We'll see.
No, we need that kind of stuff because we do. It's long form discussions, podcasts, these kind of things. Some people don't have them accessible to them, and they might not be able to retain information because they have no foundation knowledge. So having that kind of introduction to the core components is essential, ironically.
And hard to try to explain endocannabinoids in a lay term, but I'm working on it. Got some good pictures.
Yeah. One of the fascinating things about the endocannabinoids in particular is the thing you mentioned earlier about the metabolic enzymes, which seem to play a equally crucial role in the regulation of these things. So, I mean, what information do you have on that kind of thing?
On the metabolic enzymes.
Yeah, so I think is it unless I'm mistaken, it's was it fatty acid hydrolase and there was one other one which I forget.
MAGL inhibitors, yeah.
Yeah, that's
so
right.
These are okay. So when the typical THC acts on the receptor and then it just kind of stays in the receptor, it has a very different effect than anandamide. Anandamide and 2-AG act very quickly on the receptor, and then they're released. And when they're released, they're degraded by the enzyme. So anandamide is degraded by fatty acid amide hydrolase, and 2-AG is degraded by MAGL.
Yeah.
And chemists have developed inhibitors for these enzymes, which have the effect of increasing the duration of action of anandamide and 2-AG at the receptor and elevating the level. So you can boost these endocannabinoids by delivering these enzyme inhibitors. And that's, I think, one of the most promising directions for new pharmaceutical kind of drugs that modify the endocannabinoid system. So, for instance, FAAH inhibitors, fatty acid amide inhibitors, have in animal research and preclinical research are really effective antidepressants. By elevating anandamide, it maintains its effect at the receptor for up to 24 hours, where usually it only lasts for, quickly, for just a couple of minutes or very quickly. It has its effect, it's released, and then it's degraded. But the wonderful thing about endocannabinoids is they're only released where they're needed in the brain. So if you need endocannabinoids, they're produced and they have their effect in this kind of homeostatic control of of our stress system and various systems of our brain. But if you give these enzyme inhibitors, it prolongs the action for 24 hours. So you have for a few minutes versus 24 hours, a really dramatic effect of elevating these endocannabinoids, which are having effects where they're needed in, for instance, the stress system of the brain to try to reduce stress. These enzymes, they can affect depression, they affect nausea. A lot of our work has shown these, both MAGL inhibitors in the nausea-inducing region of the brain, the insular cortex. If you elevate it in the insular cortex, you reduce nausea in rats for a prolonged period of time. so in whichever system of the brain that they're needed in, you get elevation of these endocannabinoids.
You were mentioning the endocannabinoid system role in nausea and anxiety. Just before we move on to some more work that was done. So, what do you find most fascinating about those two, particularly? Because a lot of the bodies of work is based in nausea.
Right. Well, I think that our nausea work with Rafi and Roger and many other researchers was kind of a detective process in our lab anyway. We found first that if you kind of deplete the forebrain of serotonin, you don't get nausea. Rats don't show nausea in these models. So Erin Rock, for her PhD, decided to see, well, maybe CBD is acting by interacting with serotonin to reduce nausea. So she delivered it to the dorsal raphe nucleus, a region of the brain that produces basically serotonin. Neurons in this region project to all the different forebrain regions of the brain.
Right.
And so she delivered CBD to this region and found that it produced nausea. If you put it outside of this region, you delivered CBD outside of this region. We have missed cannulas that where you get it to the wrong place. It didn't reduce nausea. It had to be right in that region. Right. So, and it was mediated by 5-HT1A. If you get delivered antagonist, it prevents the anti-nausea effects of CBD. So then we started looking, okay, what region of the forebrain is doing this? And the insular cortex is a region that humans in fMRI work with when they experience nausea, that's a region that lights up. So we started looking at the insular cortex, and we delivered CB1 agonists to the insular cortex, like THC, but not it's synthetic CB1 agonists, and it prevented the nausea. And so CB1 agonists are acting, our hypothesis was that they're acting on serotonin neurons to reduce serotonin. So the way CB1 agonists work is they're inhibitory and they reduce the release of other neurotransmitters at presynapse. So we hypothesize that it's CB1 on serotonin that's reducing nausea. So we did that, and then we delivered serotonin antagonists to the insular cortex, and that reduced nausea. So the interaction. So then finally, our final experiment was microdialysis, where you actually measure serotonin in the insular cortex, and you deliver CB1, you deliver cannabidiol, or you deliver a 5-H, you deliver a MAGL inhibitor. MAGL inhibitors elevate 2AG where and when they're needed. So if we gave animals vehicle, we gave all animals a nausea-inducing drug, and that nausea-inducing drug elevated serotonin in the insular cortex. If we give the animals CBD, it prevented the elevation in serotonin. We actually measured it. We saw it prevented serotonin elevation. If we give a MAGL inhibitor, it prevented serotonin elevation. So that was kind of the final proof in the pudding that that nausea is an interaction, is produced by serotonin. And if we, in that region of the brain, not other regions, just in that region of the brain. And if we deliver CBD or a CB1 agonist or MAGL inhibition, which elevates 2AG, which is the CB1 agonist, it reduces nausea. So that was kind of over the years, the detective work we did to show pretty clearly that it's an endocannabinoid serotonin interaction that's producing this anti-nausea effect. The anxiety you asked as well. Okay, so in some of these studies, we also looked at just CBD and CBDA and their effects on anxiety, and we found that was also 5-HT1A mediated. This is with Roger Pertwee. We had this wonderful collaboration where our lab did the behavioral work to show this is what is actually happening behaviorally. And his lab did in vitro work in the test tube, and our results matched perfect. It was wonderful. We got this convergence of information showing that CBD is acting on the 5-HT1A receptor to reduce anxiety and to reduce nausea. In terms of anxiety, though, it doesn't work in a non-stressed animal. CBD nor CBDA don't reduce anxiety in an animal that's not stressed previously. The animal has to be in a stressed state. So if, for instance, if you deliver foot shot 24 hours before you test for anxiety, you get enhanced anxiety, kind of. And at the time of testing, CBDA work really well in reducing anxiety under that condition. But if you don't pre-shock them, it doesn't do anything. So that suggests that CBDA and CBD might be really effective for post-traumatic stress disorder in individuals that are in a high state of stress, high state of anxiety. These compounds might be more effective than people who just I think I'm going to go to a party and I'm a little stressed out by that. I'm going to take CBD. It's probably not going to work much. But if somebody has social anxiety disorder, and it's probably going to have more of an effect if this preclinical work translates to humans. So now we need that translation from the lab to the humans to see if it works. And that's always with cannabinoids. That's where we're missing the information. There's tons of preclinical, scientific, lab-based studies on these compounds in animals, but the translation to humans is what's missing.
Why do you think that is? Is it a difficulty in acquiring participants for the trials, or is it a lack of motivation for people specifically want to research it? I mean, why do you think there is a missing translation towards humans?
It's again the cost and basically where because it's very these clinical trials can be very expensive and time consuming, and there has to be motivation coming from some source to fund them, and I think part of the problem. But I think NIH is starting to. I mean, for the longest time, the perspective, at least in the United States, of NIH and NIDA was that there's really no useful cannabis is not doesn't have medicinal properties, and I think that's starting to change. I think certainly with the epilepsy FDA approval and such, that it's starting to change. I was hoping there'd be more clinical trials since writing the 2017 book that I wrote, but there haven't been, and I was disappointed by that as I scanned the literature. But I think hopefully there'll be more coming out.
Yeah. I mean, it's interesting that. Do you think, just obviously the influence that cannabinoids have on maintaining homeostasis inside the organism, do you think then, with that line of thinking, that we're biologically tailored to use these for our benefit?
And I'm partly because THC doesn't act just like an anandamide. It has other effects. It has side effects. It acts through it. When one takes cannabis, the THC acts throughout the brain. It acts everywhere, not just at the site where it's needed. But when your endocannabinoid system is activated and it's activated for some homeostatic reason, it's only in the site that it's needed that it's produced and released, and it has its effect. So when cannabis is taken, it acts on all the other sites in the brain, not just the site that it's needed. So I think that you have to keep that in mind when talking about it as for benefits. I think there's some benefits, and I think those benefits have taught us where to look for endocannabinoid effects. We wouldn't, I mean, the brain, where do you look for these? And so the endocannabinoids are produced. Where do we look for them? We look for them at the places where cannabis has its effects, right?
Yeah.
So that's, I think it's important information, but I don't know if we can take, I don't know if we can go as far as what you said.
Yeah, no, brilliant. That's a brilliant answer, Linda. Thank you so much for that, because that's really enlightening to hear that, because I was under the impression that it was a very specific interaction that THC had, and it was mirroring the exact actions that anandamide has, when really what we're looking for is a hyper-personalized phytochemical profile that would eventually get to that point, yeah?
Yeah, and that's where the MAGL inhibitors and FA inhibitors are having have their effects, I think.
Right, right. That's fascinating. That's awesome. So I mean, like you mentioned before, like the last project that you worked. In fact, before we talk about the last project that you and Professor Michelin worked on, synthetic cannabinoids. So synthetic cannabinoids obviously have been around for a while now. Um, they had a huge damaging effect when it was brought on through things like spice and these synthetic cannabis products and stuff like that. Do you think that synthetic cannabinoids play a role in future cannabinoid medicine, or do you think they're just exclusively used in a research capacity?
Well, I mean, I think it's more of a research capacity. I think that we're probably yeah, I think because synthetic cannabinoids, if you deliver a synthetic cannabinoid, again, you're getting these same effects as you're getting with THC. You're affecting everywhere. But the reason the FA and MAGL inhibitors are so promising is that they're only elevating endocannabinoids where they're needed and where they're produced for a purpose. So you're enhancing the natural system. With synthetic cannabinoids, again, somebody takes it, it affects all the cannabinoid receptors everywhere, and it's not specific again.
Right.
And certainly there's problems with synthetic cannabinoids. One of my final PhD students, Marika Devono, did her thesis on cannabinoid hyperemesis disorder.
Okay.
which is kind of paradoxical because THC reduces nausea, but cannabinoids are biphasic. They have an effect that's opposite at one dose than another. So a low dose of THC reduces nausea, but a high dose produces nausea. So that in our laboratory, we've shown that if you give a high dose of THC, rats will show nausea-induced behaviors. So that's so Marika took it upon herself to try to track that down. Why is it that it's producing this? And it seems to be producing stress. Synthetic cannabinoids also produce nausea in this spice. She used JWH 018, which is, I think, the component of spice. And she used Meshulam's HU210, which is a highly potent synthetic cannabinoid. And they both produce nausea in this model. And they also produce an elevation of cortisol or corticosteroid, which is a stress-related compound. Interestingly, CBD reduced the nausea produced by these synthetic cannabinoids and reduce the stress, the cortisol response produced by these synthetic cannabinoids. So CBD is again providing this protective effect, and I think that's one of the big messages. I think, at least from our work, is that CBD levels in cannabis can protect against some of these bad effects, these adverse effects produced by CB1, too much CB1 agonism.
When you mentioned earlier about that CBD was effective for PTSD, and for people that already had anxiety, the first thing that popped to mind was that I was like, oh, that must have a relationship with cortisol in some form then, you know?
Yeah. And certainly in what Marika's work has shown is that the cannabis hyperemesis disorder seems to be stress mediated. Cortisol is released and CBD reduces that. Now, this is in animals again. Again, we have to be careful. We can't say that CBD is effective for PTSD yet. We know that in animals, it's effective for PTSD models in animal research, but whether that translates, we don't know yet. Yeah.
It's so frustrating. It's so frustrating because it's like we're so close to making real
Yeah.
movements.
That's kind of this book that I keep ending at the end of every chapter is we need more research. We need more human research.
Yeah,
Yeah.
yeah, we certainly do. And I think, I mean, just for people that are unfamiliar with the scientific process, the reason why animal models are used, is it animals like rats in particular because their endocannabinoid system is most similar to ours, or is it because they're most available, or?
It's I guess because we can deliver the compounds to the rat. We can do the research with the rodents that we can't do with the humans, and
Yeah,
provide it's sort of in the process of trying to approve new drugs. There's different phases that there's in vitro work that like Roger Pertwee does, and then the second phase is animal models, preclinical work to see if the effect it produces the predicted effects in these rodent models, and then it moves to human trials to
right.
see. So it's a phase in the process. So the preclinical work is there, the in vitro work is there. A lot of evidence that cannabinoids are effective for many different things, but taking it to the humans has been slow.
Yeah, well, hopefully it speeds up in the years to come. So the last piece of work that you and Dr. Michelin worked on then, what exactly was that?
Okay. This is something that Rafi was so excited about. It was just fun. It was really fun to work with him. He pulled together a team. That's what Rafi tends to do. He pulls together teams of researchers to do collaborative work, in different fields, and to try to get a really good understanding of what this molecule is doing. So he and Aaron Lichtman at Virginia Commonwealth University, and Vincenzo Di Marzo in Italy. He's now in Canada, but I think he's in both places. He has labs in both places. Had discovered a new compound, a new endocannabinoid-like compound. It's a fatty acid amide that's released called oleoglycine. And oleoglycine is kind of like an anandamide, but it's a fatty acid amide like an anandamide. And it's released in the insular cortex, the same region that I was telling you mediates nausea, in response to brain trauma. So in mice with traumatic brain injury, oleoglycine is released in a protective way to try to reduce the impact of that trauma.
Right.
So they had discovered this, and Rafi, just in his big picture, he's always big picture. He had this big picture. He said he remembered this article that came out in Science on nicotine addiction. This patient had a stroke to the insular cortex, and he was an avid smoker before the stroke. And after the stroke, he woke up and he no longer craved nicotine. He didn't want a cigarette. It was his craving, his addiction, it was gone after this damage to this region of the brain. Some rat and mice work had shown that, in fact, if you damage this part of the brain, it interferes with nicotine addiction. So this all kind of came together. So Rafi says maybe oleoglycine is released in this region of the brain that mediates addiction to reduce the impact of addiction, to reduce the damage produced by these addicting drugs. So he thought oleoglycine might be an anti-addictive endocannabinoid-like substance released in this region. Now, I had been doing work on morphine withdrawal, opiate withdrawal in the insular cortex. Curie Wills, a PhD student with me, did her PhD on this. So he pulled me into this group with Di Marzo and Lichtman and him and me. And he said, well, why don't you look at opiate addiction and see if affects opiate addiction. And our lab did that, and we found that oleoglycine reduced morphine withdrawal responses in rats. So it was effective. And so if rats were had chronically exposed to morphine, they normally show withdrawal when you take the morphine away. These rats didn't when they received oleoglycine. Oleoglycine prevented the withdrawal. Didn't affect the rewarding effects of they still liked it. They still liked the morphine. But they didn't show withdrawal from it. So so Aaron Lichtman and Vincenzo DiMarzo are still working on this, actually, this project. But that's what we were doing just before he passed away. He was quite excited about it. Every time we met, pull the group together and say, well, okay, so what do we do next? It was a wonderful experience working with him. He developed a more stable version of it, and we tested that as well. So we went, but that's the mainstay of it, an anti-addictive compound released to reduce the impact of addiction on the brain. That's what he was working on at the end.
That is so cool and so needed as well. Unfortunately, over here in Scotland, we have the highest rates of addiction in Europe, and for a very small region, that's completely unacceptable. But yeah, that's fascinating. One of the things that pops to mind as well is that if obviously that's the insular cortex that's responsible for addiction. So one of the things is I was thinking was like they often say that a large part of addiction is habitual. So it's the action of like opening the tin or preparing whatever you're consuming and these kind of things. So is that also, do you think, in the insular cortex, or is that spread throughout? Because I have like the limbic system pop to mind, excuse the pun, where it's like these are all these different emotional associations with your addictive process.
Yeah, there's many different regions involved with addiction. Certainly the nucleus accumbens and the amygdala are involved in the rewarding properties of these compounds, and they all speak to each other. These regions.
So do you think the insular cortex just directly blocks any dialogue between these regions, and that's why it neutralizes the addiction?
Well, I mean, it's okay. It's specific for nicotine. Now, Aaron Lichtman showed that it reduces both the rewarding and that oleoglycine reduces the rewarding and aversive and withdrawal effects produced by nicotine, but it only reduces the withdrawal effects produced by opiates. So it's drug specific, and it's acting on these various on a number of different regions of the brain. I think this work has all just been systemic. So we just give the drug, and then we look at the effects on whether rats will self-administer nicotine or will show withdrawal effects from nicotine. Likewise, in the case of opiate withdrawal. So, but Rafi, the connection with addiction was because of the stroke patient. Stroke patient, the damage to this region prevented the stroke patient from wanting nicotine. So this region, it's a kind of the final way station of all of these subcortical regions that are involved in addiction. So if you destroy the nucleus accumbens, you also destroy the taking of different drugs. But it's part of the pathway leading to the insular cortex as a site for addiction. So all of it's a network. It's all a network. If you destroy the network, you'll interfere with the process of addiction.
That's a brilliant description. Thank you, Linda. That's really helpful for me. That's really helpful. So, I mean, we've essentially covered most of the things I'd like to discuss. Just two more things, really. What do you personally find most exciting about the work that you do?
Well, I guess the most exciting was this detective work we did with nausea to try to figure out how endocannabinoids might be interacting with where they're acting, and discovering that it was their interaction with serotonin, and the site of that interaction being, again, the insular cortex. It's actually a region. The insular cortex is a big region. There's the visceral and the interoceptive and the gustatory, and there's different regions. And ours is it's this interoceptive insular cortex. If you have cannula in a different part of the insular cortex, it doesn't work. It has to be in this specific region of it. And it's that discovery process that has kept me going and kept my students going with excitement throughout the years. Yeah.
So where do you see the research walking towards in the future? Like, are you aware of anything that's like primary and relative, like coming soon, or I mean, what do you see happening in the future for cannabinoid research?
Well, I mean, I think it's sort of unlimited. I'm not sure. In terms of this oleoglycine work, I think Aaron Lichtman and Vincenzo DeMar. Aaron Lichtman is continuing to work with different with nicotine addiction, trying to pinpoint the mechanism, the site and the mechanism where this is occurring. And Vincenzo DiMarzo is working on feeding, kind of the effect of oleoglycine and oleoalanine, these compounds that Rafi discovered on feeding behaviors. And so in terms of this research, that's where that's going. I think there's all kinds of research going different places. I actually don't have a lab anymore because I'm retired. So,
well,
so I'm not playing a role anymore in discovery, which is kind of sad.
Well, to be honest, it's evolved into this, Linda. It's like you're playing the role now. The people that are watching this are going to be greatly educated by you coming on the channel, and the information you're providing about Dr. Michelum and all the other things you've talked about today has been fascinating. It's no, thank you. This has been fascinating. But that's where it is. You should make. I mean, obviously, I have no idea what your personal life is like, but please, like you should make your work more known. Get on podcasts because this has been brilliant. This has been really good.
Thank you. That's really kind of you. Thank you so much.
Well, you're very welcome. Well, I think we've
Fun to
essentially.
talk to you.
Sorry?
It's been fun talking with you, Colin.
Yeah, I've really enjoyed it. I've really enjoyed it. I mean, before we go, is there anything you'd like to add?
No, I don't think so. I mean, I think you've touched upon everything, and I wish you the best with this podcast. I hope you get. You might want to try to reach out to some of the other. I mean, you have. I know you do. And there's others you might want to reach out to.
Well, if you've got any names, please feel free to send them over because I will literally email every person you send over. Honestly, I mean, sincerely, if you do. But yeah, thank you so much for this, Linda. Yeah, that'll do us for today. Thank you so much for your time.
Okay, thank you, Connor. Good work, and it's a good show.
Thanks to Linda for giving me her time, and thanks to you for sticking around until the end of the video. I sincerely hope you enjoyed the content. I'll see you next time.